Right, the question that we want to focus on this time is looking at different diseases and different ways that things defend themselves, including the immune system. Right. Around this, within the specification, there are a few different pathogens, a few different diseases that you need to know. Don't put off knowing them because there is a good chance that they will come up in the exam. So the question starts off with rose black spot disease is caused by which type of microorganism? Right, the answer is a fungus. Right, 9 times out of 10, if you're coming up with something that's going to cause plant disease, 9 times out of 10, it will be a fungus. Right, part 2, it says explain how different types of organisms defend themselves against microorganisms. Right, that question in itself is a bit ambiguous, right? There are a range of different things that you can go and talk about. The fact that it's talking about types of organism clearly states that you need to talk about more than one type. Personally, I would talk about both plants and humans. Right, remember some of the ways that plants go and defend themselves. They produce chemicals that will go outside of the plant cells and go and kill the microorganisms. They'll go and produce extra chemicals that go and make the cell wall of the cells harder, so it's more difficult for pathogens to get in. You can go and talk about the fact that they'll go and produce chemicals that will go and alert other cells that there's some sort of microorganism invading them and that they need to get the defences up and running. Right, if we then go and look at humans, you can talk about different defence systems. So you can go specifically talk about white blood cells, so your active immune system, talk about antibodies, you can talk about phagocytes, which go and eat microorganisms. You might talk about physical barriers, such as the skin. You might talk about stomach acid killing off pathogens, all manner of things like that. So there's a large list of things that you can go and include. Don't get flustered by the fact that it's talked about different types of organisms, and don't sit there and just think, oh, I don't know any different types of organisms, right? Primarily, it'll be talking about humans, but you might also want to go and chuck in a few bits about plants as well. So the next part says that students try to grow bacteria in the laboratory. Figure 3 shows some of the equipment used. Right, this is one of the required practicals that you should have done. Right, so it goes and it gives you the method. So it talks about remove the lid from the Petri dish, remove the lid from the bottle containing the bacteria, use the inoculating loop to go and inoculate it, spread the bacteria over it, put the lid back on, incubate 25 degrees for 24 hours. So the question goes, steps 1 to 5 could cause a sample of bacteria to be contaminated. Give 3 improvements to the method. Right, this is where you need to think about what you've been taught about aseptic technique. So just keeping things sterile. The ways that you do this is that the inoculating link, you sterilise it, so you dip it into alcohol, you burn the alcohol off. You only go and take the lid off of anything when it's underneath a Bunsen burner flame, because that Bunsen burner flame will go and kill any sort of pathogens that's coming down and landing on it. You might want to go and talk about sealing the lid onto the Petri dish in 4 places so that it doesn't come off. You might want to go and talk about just gently flaming the top of the bottle that the bacteria is growing in, right? So it all focuses around that aseptic technique. Right, the final part of the question says, why do students grow bacteria at 25 degrees rather than 40? Any bacteria that will grow at 40 degrees will more than likely be able to grow inside of us. In the same way that any bacteria that can grow without oxygen will be able to grow inside of us. Right? So they're the key things that you need to be thinking about when answering this question. Right. Go back, watch any part of the video again that you're unsure about, get your biology specimen paper and have a go at answering this question.